Biology Part 19. The molecules of DNA are built up from nucleotides containing the 5-carbon sugar deoxyribose, and they are always double-stranded as illustrated in the figure. The true strands are fully complementary, so that the entire molecule, which may contain millions of nucleotides, is held together by the base pairing. At the same time, the molecule is twisted into the double hells of configuration. The full elegance and compactness of this structure is called Watson-Crick model of DNA and can be appreciated from the figure, which shows the position of all various atoms in a short length of DNA. This structure was first determined by the analysis crystal of DNA using technique of X-ray crystallography. The significance of the model is that it suggests a mechanism whereby new copies of DNA molecules can be produced. Because of the two strands are fully complementary to each other, they can act as a template or mold for making the other. With the aid of an enzyme called DNA polymerase, DNA strands can copy themselves or replicate as outlined. The process illustrated is called semi-conservative replication because half of the original parent molecule is retained and conserved in each of the new strands produced. Replication is essential so that the cell can provide identical copies of its genetic illustration, and it is to be passed to the daughter cells during a normal cycle of cell growth. DNA doubles so that when the chromosomes separate at the cell division, each daughter cell receives genetically identical package. Further details of cell division are given in the unit as well. DNA and chromosomes. The total length of the DNA molecules in a typical mammalian cell has calculated to about 2 meters, giving a staggering total of more than 10 billion kilometers of DNA in the human body as a whole. Each chromosome in an individual cell is believed to contain just one single long molecule of DNA, so that 46 chromosomes of a human cell correspond to 46 individual molecules of DNA, each about 4 centimeters long. To prevent the interior of the molecules from becoming a hopeless tangle, it is necessary for the DNA threads of individual chromosomes to be coiled in some organized way. This is the function of the specialized histonic proteins which are associated with DNA, and histones are now known to form particles called nucleosomes. Nucleosomes are also formed there. These have the structures shown in the figure, and each consists of a length of DNA and double helix containing about 200 base pairs of wound around the bead of historic proteins. In an activity growing cells, the chromosomes in the molecules are mainly present as along the chains of the nucleosomes. However, when the cell is preparing to divide, the chromosomes coil up much more tightly and become visible by a process called, there is a process that we need to mention here, and it makes it visible. Well, in the previous saturation, of course, it was not visible, and there was a stamp on it. So this is what we call condensation. This is partly controlled by interaction between the histonic proteins of the individual nucleosomes and partly by the histonic proteins